when I heard about environmental forensic, I was basically thinking about CSI, how cool I would be as a forensic investigator.
Right, so this is the basic workflow of data analysis of any data set, not just environmental forensic data set. First, we collect the data, and then
basically this is your cell. So inside your cell, there's so many things, but the most important thing is the nucleus. The nucleus contains all your genetic information.
Okay, good evening, everyone, and welcome to tonight's Zoom webinar on a topic that is promises to be very interesting. It is on environmental forensic theories and application. Okay, and if you were to Google and to find out what environmental forensic is a very simple definition, is the application of defensible scientific methods to address questions related to release histories and sources of contamination in the environment. Iaitu forensik persekitaran adalah aplikasi teknik sains untuk menjawab soalan-soalan mengenai punca dan pola penyebaran bahan-bahan pencemar di persekitaran. Okay, the question that arises from a person's mind is why environmental forensics? Why is it important? Kenapakah bidang ini penting dalam perlindungan alam sekitar kita? Ya? Well, alam sekitar kononnya dihadapi masalah tragedy of the commons, di mana individu-individu menggunakan sumber alam secara bebas bagi kepentingan diri tanpa mengambil kira kebaikan penduduk atau rakyat jelata. Ya? Dan kemerosotan sumber dan persekitaran yang dikongsi umum yang berakibat daripada tindakan kolektif mereka ini dianggap sahaja sebagai collateral damage. Ya. Siapakah yang juga menanggung kos kerosakan persekitaran kita ini? Sudah betapa kerap dan lamanya berlaku pencemaran terlepas sahaja daripada hukuman dan masyarakat umum yang terbeban dengan gejala buruk akibat daripada perlakuan mereka ini. I think if you think back on the kinds of environmental disasters that we face, for example, the Sungai Kim Kim pollution that affected a lot of people, communities living around the area. And so forensic scientists will help to ensure that those who contaminate the environment are found out and are discovered and brought to justice. Okay. So bidang forensik memang ada peranan detektif ya dalam isu persekitaran. Ya. Bidang forensik persekitaran membekalkan asas saintifik yang dapat bantu menentukan dengan lebih tepatnya punca pencemaran alam sekitar, bila pencemaran mula berlaku, sejauh mana ia disebarkan dan setakat mana terdedahnya masyarakat umum. Penyiasatan ini seolah menandakan cap jari atau fingerprint atau tanda tangan, the signature, pencemaran supaya pihak yang bersalah lebih senang dibongkarkan. Okay. Malam ini memang kita bertuah kerana ada 3 orang pakar dari USM yang sudi meluangkan masa untuk berkongsi dengan kita semua pengetahuan dan pemikiran mereka tentang bidang forensik persekitaran dan bagaimana ia dapat menghasilkan bukti-bukti ukuh yang dapat diguna dalam pembicaraan apabila perlu. Speaker-speaker malam ini akan tumpukan kepada 3 aspek penting. Ya. Pertamanya, Dr. Syahidah akan she will talk on techniques. She said not biochemical is chemical or biological techniques, not but chemical techniques for fingerprinting pollutants. And she'll be followed by Dr. Yusri on data analysis and interpretation to detect patterns of contamination. And then Dr. JediPenang Lalong will talk about microbiology tools for toxicology detection. Okay.
Again, thank you very much. And Assalamualaikum and a very good evening to everyone, especially to the listeners out there. Thank you very much for your participation. So I will start by sharing my screen. Okay. So I hope everyone can see my shared screen clearly. Right, Dr. Khan has introduced my educational background. So I'm just actually trying to probably just give an extension on the research area and expertise. Okay, and that what I have been involving in the university or USM. So basically, I have been involved with chemical fingerprinting in environmental forensics. I did my PhD with using stable isotope profiling, and then during my masters, I was involved in the environmental monitoring of organic contaminants. So basically, my research experience has been almost involved every time with regards to environmental chemistry. Okay, I'm attached to the School of Industrial Technology as well as a research fellow at the Analytical Biochemistry Research Center, where all the expensive instruments in USM are. Okay, so I can be reached at Syahidah.Muhammad@usm.my. Okay, so that's basically a bit of introduction from me. Right, environmental forensics. So basically, when it comes to environmental forensic, the main purpose of this branch of science is to answer the 4 W's as well the 1 H questions, such as who caused the contamination and what are the contaminants out there, where did it come from, when did it occur, and how did it happen, whether was it did it happen by accident or did it happen intentionally, okay, by the culprits out there. Okay, so if we can answer all these questions, then we are basically on the right path with regards to our investigation. Okay, and to tell you the truth, from what I know, the disciplines in environmental forensic is basically covers many branches of science. There's the program that I know from I think offered by the University of Bangor in Wales. They are basically offering environmental forensic programs for undergraduate study. So if you can imagine, if environmental forensic can be offered as a program, then there's such a vast knowledge involving with this branch of research. So, but the thing is, what we offer in TI only environmental forensic as a course. Okay, so we have to cut out many of the disciplines. Okay, but we still really focus on the main disciplines in environmental forensic, which is chemistry, statistics, and biology. Okay, all the other disciplines are basically complementary in terms of fulfilling the investigation in environmental forensics. Okay, let me talk a bit on the origin or the birthplace of environmental forensics. There's a big oil spill cases way back in 1989. So it involves the major oil spill from the big tanker, the Exxon Valdez in Alaska. So back then, it was the biggest oil spill in the world. But in 2010, it has been surpassed by the Deepwater Horizon case. So, but still, in terms of volume, the Exxon Valdez oil spill is basically up there, okay, in terms of volume. So when this oil spill occurred, people have been really talking about fingerprinting, oil spill fingerprinting. They want to actually investigate how far the pollution happened, or how far the traveling of the pollutants. So then, because of this issue and the big controversies surrounding this oil spill, Dr. Robert Morrison is basically the father of environmental forensic. He was the first one who coined the term environmental forensic, especially in his book and many other of his publication. So it's actually a quite recent or quite a new branch of science. When I heard about environmental forensic, I was basically thinking about CSI, how cool I would be as a forensic investigator. Okay, so with all the sophisticated instruments involved, but after some time I realized it's not as rosy as CSI made it out to be. Okay, for an environmental forensic, it's basically this: it's dirty and it's tedious, it's laborious, and basically at times dangerous. So these are the pictures that are involved when you think about environmental forensic investigation. Okay, okay. Right. So now I would like to go deeper into the area that I've been involved in environmental forensic. So it's basically my expertise, which is the chemical fingerprinting. Okay. So how do you actually identify the chemical fingerprints? Okay, the first one, I would like to talk about hydrocarbon fingerprinting or hydrocarbon profiling, because as you can see, petroleum or petroleum products are basically one of the most ubiquitous pollutants out there. Okay, so people have been doing hydrocarbon profiling a lot, okay, to investigate the origin of the contaminants with regard to hydrocarbon. So to the layman, hydrocarbon that are present in petroleum product, they are basically the same. Or if you think about petroleum product, in terms of appearance, yes, there's a bit different. But when you think about chemical constituents, basically most of them, they think that is kind of the same, but it's basically not. When you analyze it with a specific or with a sophisticated instrument, you will find that there's a bit of difference in terms of the hydrocarbons profile, okay? When you analyze using the instrument. So basically, if you look at this, I'm not sure whether you can see the mouse, okay, moving. Basically, when you have the hydrocarbon profile in relation with the petroleum product, you can see that for jet fuel, it has a bit of a light chemical constituent in it. So they basically have a specific hydrocarbon profile. But if you look at diesel, diesel contains middleweight chemical constituents or hydrocarbon. So it basically have a different set of profile from that of jet fuel. Okay, so if let's say you want to investigate an oil spill in this near the seashore, for example, you cannot really, based on the appearance, think of, oh, this is probably diesel, or this is probably jet fuel. So it doesn't work that way. You really have to analyze it in the lab using sophisticated instrument, and from the profile only you can basically prove in court that these are actually diesel product, or it comes, or it's probably a bunker fuel, for example, because if you can see here, bunker fuel or heavy fuel oil, they also have different type of profile when it comes to hydrocarbon constituents, okay, inside the petroleum product. And if you look at the last two figures, it shows the heavy oil or lubricant oil. So these are also very different from the other petroleum product. So if you look at this hydrocarbon profile, they are basically can give you an idea, okay, what are the petroleum product out there, okay. So that's what I have to say with regard to hydrocarbon profiling. Okay, so now we move on to another type of chemical fingerprinting, which is the stable isotopes fingerprint. So stable isotope fingerprints are basically DNA fingerprints to the chemist, okay? So it basically gives a signature to any samples or any samples that we analyze. So because these samples has a specific isotopes, okay, inside the samples, okay, the chemical makeup. So based on this profile of the stable isotopes, you can actually differentiate the samples, okay, whether, especially on the origin of the samples. Basically, stable isotope can provide you the history of a sample, because stable isotope will change every time there's a, what do you call that, there's a reaction going on with the samples. So anytime this change happen, it will be stored in the chemical makeup of the sample. So with stable isotope fingerprint, you can determine the origin. You can basically identify the dietary habits. If you analyze the samples that is obtained from organisms, for example. So basically, it can tell you many things. Okay, I cannot really go deep into all this, what do you call that, this fingerprinting technique, because it may sound like I'm giving a lecture to my student. So if you want to know more about all this fingerprinting technique, you can actually drop me an email, okay, or have a private discussion with me. And so, and another example with regard to the stable isotope fingerprinting, you can see here that as an example from the book that I take from from Stephen March in Methods in Environmental Forensic, this isotope ratio fingerprint can actually resolve 4 gasoline into 2 groups of 2, which are derived from different crude oils. So basically, if this crude oil, if it comes from the Middle East, then it can basically be differentiated from the crude oil that comes from another part of the world. Okay, so the differentiation in the stable isotopes of the crude oil can actually be detected. Okay, and the pattern are quite different, and basically this fingerprint, what do you call, information can actually help us in differentiating the geographical origin of the petroleum product. Okay. After stable isotope fingerprinting, I'm going into trace element profiling. So basically, trace element profiling are the inorganic elements, okay, whereby they are actually ubiquitous in many samples, for example, in soil samples. So if you look at the figure here, these are trace element profiles in soil samples from different locations in Australia. Okay, so if you look at the graph or the chart, you can actually look at the differences in the trace element profile based on the concentration. Okay, so see if trace element profiling can actually differentiate the geographical location in a country like Australia. You can imagine the differences that this trace element can represent if you take the samples from different countries even. So it basically is a great tool to differentiate the origin of samples. So it can basically help us in our forensic investigation, okay, in determining the geographical location, okay, of the sample that we have. Right. But okay, now I'm what do you call giving some continuation from the multi-element profiling just now. With regard to this graph, is basically using a simple Excel, okay, just to look at the differences in the concentration. But if you imagine if we have thousands or hundreds of samples, then with our very own eyes, we cannot really differentiate it because there are too many samples to actually detect or tease out the differences. So that is why we actually need multivariate statistical analysis to look at the differentiation of the samples. Okay, so basically, if you plot the data using multivariate statistics rather than just simple statistics, we can actually differentiate all the samples based on the trace element profile, and you will have clusters, okay, like this, whereby it will show you the differences of the origin of a sample. So this is an example of the wine samples that can be sourced from different regions in Australia. Okay, so basically, they want to detect the what do you call the real wine samples from Coonawarra or from a certain wine region in Australia, because there are some claims that the adulterated or confiscated wine, premium wine, they claim that it comes from this premium wine region, but basically they are actually not. So if you look at the X samples, okay, the samples denoted by X, they are actually come from Barossa Valley, which are what do you call it, which are not really the region that they claim to be coming from. So basically, these are one of the helpful tools that multivariate statistics can offer. Okay, and there's also another example that how trace element can actually be helpful in differentiating the geographical location of a sample. So, for example, I have here the plot of the of rice samples coming from different countries. So, if you look at the sample denoted or colored in purple, so these are the test sample, whereby we want to know the origin of the test sample. So if we plot the trace element using multivariate statistics, you can see that it falls into a specific cluster, which is the Thai country cluster. So it basically, what you call it will basically helpful if you combine the multivariate statistical technique to tease out the information given by the trace element concentration. Okay, so any sample you can actually make use of the trace elements which reside in it, and you can basically differentiate it or discriminate the geographical location of the sample, which are very helpful for your environmental forensic investigation. Okay, so I think basically that's the end of my short presentation, and I think my colleagues can help me to basically what you call that, explain more on the other branches of environmental forensics. Thank you very much.
All right, can you hear me? Can I have an audio confirmation?
Yes.
Okay, thank you very much. And thank you to Dr. Kam for introducing me so graciously, and Dr. Syahidah for introducing the topic, because she laid a lot of the groundwork for the topic that I'm going to talk about now. And it makes my job a lot easier, because you have some backdrop of what of the environmental forensics, the background of environmental forensics, and also the data that's collected in environmental forensics. And then I swoop in and talk about the analysis, the data analysis part of environmental forensics, which is the second half of what Dr. Syahidah does as a chemist. So data analysis is like I mentioned, it's actually the second part of you go to the of an environmental forensic investigation. You go to the field, you collect your samples, and then you bring back to the lab, and then you run a few tests on them, and you collect parameters from the samples. And usually, when you start off collecting samples, when you collect samples and also you analyze the samples, you basically you try to run a slew of tests on the samples to get as many parameters as possible from the same samples. And sometimes if you're lucky, then you'll be able to come up with some fingerprints, or like Dr. Syahidah mentioned, some fingerprints or some unique characteristics of the sample that makes it easily separable between other samples. And that technique on how to separate the samples according to this unique characteristic is what data analysis is for in environmental forensics. So basically, in data analysis for environmental forensics, we'll be finding patterns in the data. And sometimes it's not so obvious. It's not as easy as just running the first test and running the first analysis and you find the pattern. Sometimes you have to experiment a number of tests, a number of analysis to get those patterns. So I'll give a few examples here. All right, I'll just move this a bit. Okay, now the techniques that we use to analyze the data is elaborate sometimes, and it doesn't lend itself well by using Excel. I'm sure most of you know what Excel is as a spreadsheet software, or other spreadsheet software. So I think the most popular is Excel. It's because there's a lot of different techniques or different methods used within a single technique or single data analysis method. And if you can actually do it in theory in Excel, it's just that it can be quite difficult because of the many steps that you need to take. So to make it easier actually to run all these complicated data analysis techniques, we would use like specialized software. For example, R is a very popular data analysis tool, not just for environmental forensics, but for other data analysis projects also, and also modeling work. And I think now as Python is actually very popular. I know Python is more towards programming, but you can actually, when you actually run data analysis on these very large data sets that comes from environmental forensic investigation. So inside Python itself, there's a lot of packages or modules that can help you analyze your data easily. And aside from that, MATLAB is also quite popular to analyze data. As you can also run modeling work, but yeah, it's a licensed software, so that makes it a bit less popular as these two, because these two are free. So the things that we do when we analyze data are these three things. The comparative techniques in statistical methods is by it's just basic statistics. We want to compare whether one average is higher than the other average. For example, you collect samples at site A, and then you run tests on them, and you get the concentration of a certain chemical at site A, and then you get a mean, you get a value, like say 10 micrograms per meter cube, and you go to site B and you got it at 9 micrograms per meter cube. Then you want to compare, is it really this 10 micrograms per meter cube, is it really higher than the 9 micrograms per meter cube? So you need to run statistical methods as the techniques on them to show how much different they are, how statistically significant differently they are between these two, before you can conclude that they are actually one is higher than the others. So that is basically what comparative techniques is about. It's just comparing them and then giving a quantitative value on how much different they are. Maybe you are confident that this is site A is higher than site B by 99%, or maybe 90% if you're less confident about how different they are. So basically that's what this is about. And the second method here, multivariate methods, or okay, I'll talk about multivariate methods, is something that has been introduced by Dr. Syahidah. Basically, it's about analyzing the data that has many variables, hence the name multivariate or multiple variables. So when you collect samples and you run tests on them, and for example, a multi-element test on a sample, and then you get many parameters from that one test. For example, you get the concentration of potassium, you get the concentration of sodium, you get the concentration of calcium, and so on. So you get many parameters, and these many parameters are the many variables. You want to analyze all of them at once. You call it a multivariable or multivariate methods. Now we run this. It's some sort of a series of steps of analysis that that tease out, like what Dr. Syahidah mentioned, tease out the patterns in the data that we cannot see with the naked eye. But the good thing about this method is that it's fairly automated. So once we've already arranged the data accordingly, we can just run through the algorithm and it will be able to give us the pattern automatically. Now, recently, not that recent, but maybe for the past 10 or 20 years, there are these machine learning techniques that can also take out the patterns in the data, such as neural networks. If you're familiar with machine learning or artificial intelligence, those things can also tease out patterns from the data aside from multivariate methods. But the most common right now, the most very standard methods for us to use when we analyze these multivariate methods are the multivariate methods, like just now Dr. Syahidah mentioned PCA. Now, the last technique is geostatistical techniques. So it's again, in the name, there's a statistical, means that we use statistics to differentiate two different samples, but then we superimpose that on maps. So hence the word geo there, or the prefix geo. So like when we look at data, then we see them in 2 dimensions. That means we see the rows. I'm going to show that slide later. So we see the rows of data, and then we see just on that spreadsheet. But then when we apply geostatistical techniques, we get the data, and then we map it out onto maps, onto areas you see from the top, and then you can visually see how the pollutants, if you're talking about pollutants, span across an area. So that is a combination of statistics and also the geo part. Okay, so that's basically the 3 things, the 3 statistical methods that we employ in environmental forensics. Now, these things are not just for environmental forensics. We can actually use it for any other methods, any other fields. It's just that if you want to give credence or give some weight towards your analysis, you need to apply statistical methods, because through the statistical methods, you'll be able to quantify how confident you are that a certain pollutant comes from a certain area. And this is actually very crucial, because once you can say that a certain pollutant comes from site A, then you can say that maybe at 90% or 99% you're confident. So there's still that 1% uncertainty there, but at least you have some quantifiable way of saying how certain you are. Okay, let me go to the next slide. Right, so this is the basic workflow of data analysis of any data set, not just environmental forensic data set. First, we collect the data, and then we combine. Now, from my experience of working with some of the samples, not samples, but then data collected from the field, and then they bring it to the lab, and then they analyze it, and they give me the results. From my experience is that most of the work comes by collecting this data and then consolidating the data, because usually the data will be messy. You'll get it from site A, B, and C, and then sometimes the specification of instruments used for site A, B, and C are different, and then you need to like standardize the specification, then you need to merge the data, and then you need to like basically consolidate the data. So that actually takes a huge chunk of the data analysis time. And after you've done that, after you merge all of the data, making sure that they're all standardized, because you don't want like one area has a better accuracy than the other. In the end, you have to go for the lowest accuracy level, because you have to standardize everything. Then you go for quality checking the data to see if there's any missing data in it, if there's any improbable values in it. And sometimes we can do it visually. Sometimes we can run algorithms on the data to remove the bad data, and that goes into the data cleaning part. Now, most of the work of data analysis on the environmental forensic data set is this part here. I think 80 to 90% of the time is spent on consolidating, quality checking, and data cleaning the data before we actually run any analysis. So that's why it gets tedious in this part here, because when we run data analysis work on this very large data set, sometimes it can be very overwhelming because of the huge amount of rows that you need to work with. Like say sometimes you go a few thousand and even to a few million if the sensors or the samples are automated. And then after you're done, after all that tedious job is done, then you go towards visualization. Now, when you go into visualization, you actually start off with any visualization. You start out with a normal line graph, a bar chart, or a box plot, or all these things that we check out the distribution. And then we hope to see some patterns in the data from there. And then we run the exploratory analysis. This includes the multivariate statistics and so on. Now, once you get this result here, most of the time it's not going to be that clean. It's not clean means that it's not going to give you directly the answers that you're trying to find. Like say you want to discriminate the samples. Most of the time you find that you're not able to discriminate it, and then you find that most of the parameters doesn't give this distinguishing feature that able to discriminate the samples. And you go back to the drawing board in the beginning to actually collect the data, or maybe look for other sort of parameters to actually discriminate. So you go through the entire process again to go back here until you actually get that discrimination factor. Discriminate means that to differentiate the samples, because there's no point where you collect everything and then every samples can be grouped within together, one group, and then you're not able to know which is which. So to actually know which is actually to get the correct parameter, or the correct, not really the sample, but the correct parameter to differentiate between the different parameters. So yeah, that's the quality, that's the trial and error part of here, especially for environmental forensic, because okay, you go to the environment and you collect samples and you measure you run like a standard test, check for elements in the samples, but what if the elements that you actually check doesn't show that distinguishing feature, and then you need to run the samples again with other elements so that you'll be able to differentiate the samples. So and then you go through again until you can find that differentiating factor. And then we develop the model to run predictions on it, and then we write the report. So if you notice that this part of the work is actually like a huge chunk of the data analysis work. Next slide. Okay, so this is real data, but it's a textbook case data. Maybe some of you recognize this already because I recognize some of the names in the participant. So here we have okay, when you, for example, you go to a site, like say we call that a car site, and then you collect a sample and then you run it through the machine that gives you all these parameters. Okay, so you have a lot here. You have like 1, 2, magnesium, phosphorus, potassium, calcium, so 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, and then you get these concentration values for this sample. Then you sample again and you get another set, and you sample again, you get a sample again. And then hopefully from all these elements here and all these samples, you get some sort of fingerprint or signature for this site. We don't know yet. We just go there, collect samples, and we run the analysis. We just see this for now. And then you go to the next site, and then you collect samples, and then you run the test on it, and you go to the next site, you collect samples, you run tests on it, and you get this. So obviously, you think that you'll be able to know which is which, right, from here. So this is what you would see, and this is just one part of it. You cannot see with the naked eye. You can still see the numbers, but you cannot see the patterns in the data just by looking at this. It doesn't magically tell you where a certain sample comes from. For example, in this case here, we see a pollutant at scene. So we want to determine whether the pollutant at the scene actually comes from the plant or comes from the car. So that's the question actually. So the question, are these the patterns of values here? The values here, does it have a feature that is similar to either plant or car? That's the question. But just by looking at this, you're not able to tell whether actually the scene is coming from either car or plant. So we start off with visualization. If you remember, we look at some of the trends. Okay, some of, like say for example, zinc. We get the concentration of we plot it out and see how it behaves, and we see for the car is actually this value here. Oops, sorry. And then for plant, we see that it's quite a heightened value, and then for scene is a different value. So even from here, you can't really differentiate between the three things because it looks the same. For example, this one and this one looks the same. Well, this one, it looks a bit higher, but still basically the same. But you look at another parameter, then you can see that these two are actually a lot different in here. So this gives us some hope that maybe these two things, this particular element is able to differentiate between the three things. And that's what I mean by what is the factor. If you are able to, you're lucky enough to get the element that the parameter that can differentiate, for example, like this, then there's hope that when you run the analysis, you'll be able to distinguish the samples. So this is the result. So I'm going to go straight. This is actually two ways for us to run the analysis, to run the exploratory analysis, to differentiate. This is a cluster analysis, where you can see that it's actually is able to separate the different sites, but there's still some overlaps. You notice that here we have plant, scene, car, and scene. Plant is one group here, and another group here, car and scene, and then so it's still not able to effectively distinguish between the different sites. But when we run PCA, we can see clearly. You've seen this inside Dr. Shahidah's slide. You can see that the scene is actually overlaps with the car, right? And then the plant is obviously away. It's not within the same category as the car and the scene. So this gives us some ideas. This suggests, or at best we can say suggests or indicate that the pollutant that's at the scene is actually from the car itself. And then we get this, and this is how we say that, okay, the pollutant is coming from here, from a certain site. Now this is for plant, scene, and car, but we can also apply this for site A, site B, site C to determine the source. And this comes from that this data set, right? By running through those analysis. Now this is another example of the origin of honey. This now Dr. Shahida was talking about the origin of rice. So different geographical locations will have different characteristics. They will produce different characteristics in the food that's produced at that area. So by right, if we collect enough parameters or we collect enough samples, I think not enough, the sample is not that important, but enough parameters here that you measure that be able to distinguish between the samples, then you should be able to separate it, just like the car, plant, and scene samples case just now. So this is the data, and this is the results for that those different types of honey. So you see that this is less apparent, not as clear cut as the plant, scene, and car, because the cars and scene is a textbook sample. So that's why it gives such clean answers, clear results. But once we do it for another sample, we see that at 90% confidence level, we see that it's not so easily differentiable between the different types of honey in the area. Yes, we can see this is clover, this is manufacturing, this is kamahi, manuka, these are the different types of honey, but they still overlap. And then we cannot say for certain that they are not the same, other than that it's labeled by as manuka, kamiha, manuka. So probably at 90% level, for example, this case here, it's by all sense and purpose, it's the same type of honey. But at least we can say that the honeydew honey type, this category is separate from these two. So even for this case, it's still differentiable. We'll be able to see the difference. But in most cases, when we go to the site, we collect samples and we do the test. Most of the time, we don't see any grouping at all. We just see it as one group, and then that doesn't help at all in differentiating. And then we need to go out and sample again and run different tests to find that parameter that be able to separate the sample so that we can get something like this. Okay, so that is basically what we do. So of course, there are different techniques. This is just like straightforward techniques down to this, but sometimes there are many steps where we need to pre-process the data. We need to clean the data. We have to transform the data and so on before we can actually get to discriminating the samples. So that's it for me. Okay, the last part is actually the geostatistical techniques. So this is what I mean by we collect samples and we map it out onto the map, and then we apply smoothing functions on it to actually come up with areas of higher concentration. So for example here, this is not actually smooth. You can imagine like we collect samples here, we collect samples here, we collect samples here, around this area, and then we run an algorithm on it to smooth it out, and then this is what it says. So the concentration, the likelihood that the pollutant comes from a certain area will be shown by this algorithm. So it's something like the previous analysis, is just that it's applied onto a map, and that's the geostatistical part. And the technique they use is kriging. I think this is a very popular technique to actually smooth the values onto this so that it comes up with this contour plot. So those are the three basic techniques of environmental forensics, but those are the main ones. So there's a lot of modifications that need to be done on those techniques before you can actually determine the source of a pollutant. Okay, so I think that's it. I hope that clears up some of the points on how to run data analysis on environmental forensic data sets. Okay, then head it back to. Yes, I'm done.
Thank you, Dr. Yusri.
Yes.
We have the third speaker who will come on, and that's Dr. Japaring Lalung, who will talk more on the microbiological aspects of how the kinds of microbiological tools that can be used for toxicology detection.
For the introduction, and thank you very much to Jedi, and also Madam Kusalma and Izudin and Dr. Kam and everyone else for inviting us tonight. We really appreciate that. Is my sound quite clear?
Yes, you're fine.
Okay, thank you very much. I'm going to share my slides now. I can see a lot of my students here. I hope they're not going to ask me about the exam questions. Okay, let's see. Let's share the files. Okay, so basically, Dr. Syahidah and Dr. Yusri and I, the three of us are actually teaching environmental forensic subject in USM. And how do I go to the next slide? Okay, so basically we teach environmental forensic at 2 levels in USM. One for undergraduate, a master's level by coursework. It's a 1-year course, and also a Bachelor of Technology in environmental technology. It's 3.5 course, and we teach environmental forensic in 3rd year. It's an elective course. And luckily we are doing this today because 2 days ago there was a test on environmental forensic paper, and the question was how to detect presence of coronavirus from wild animal. So I hope my students know the answer, and I hope by the end of this lecture we'll be able to get some ideas. Please forgive me if it is going to be quite complicated. I'm trying my best to make it as simple as possible. Actually, this is quite simple, I hope. So let's go. I have another one to see the next slides in this phone so that I know what the next slides is. Okay, so I think before we started the DNA part of environmental forensic, I think we need to understand the real basic of DNA, because for DNA forensic or DNA technique of detecting anything, you either know it or you don't know it. You either know once, or you either know all or you know nothing, because we need to put everything into perspective before we're able to actually do it. So like when we train the students in the lab, normally we have to make sure that they really know what they're doing, because otherwise they will do like 10 mistakes, and if you do 1 mistake, you're going to do things wrongly. So basically, because we are dealing with DNA, everything has to be precise, and almost no mistake is acceptable. So I'm going to start with very basic things about cell, nucleus, chromosome, gene, genome, DNA, RNA, nucleotide, base, transcription, expressions, ribosome, translation, amino acid, polymerase chain reaction, RT-PCR. These are quite complicated, but actually it's very simple. Again, like I said, for you to be able to do the DNA forensic, you have to know all of them. If you just know 1 or 2, then it's not going to be possible. But actually, all of them are quite simple. Let's try to do it together. If you notice when Dato' Nur Hisham talked about detection of COVID-19, he keep on talking about RT-PCR or PCR. I am going to try as much as possible to explain a bit about PCR here. But let's start with cell. Basically, this is your cell. So inside your cell, there's so many things, but the most important thing is the nucleus. The nucleus contains all your genetic information. So basically, the nucleus, if you dissect the nucleus, you actually will see that there are chromosomes inside it, and then this chromosome contains all your genetic information. Actually, if you extend chromosomes, if you break them down, you will see a complete genome. So this complete genome contains all your genetic information. So from nucleus, it has chromosome inside it, and inside the chromosome we have genome, and this genome contains all your genetic information. So basically, what you're able to do, what you're going to become, is actually depending on the genes that you inherit from your parents. So if you don't have the genes for blue eyes, you will never have it. If you have certain genes for anything, then only then you can produce the protein. Only you can have the trait. If you don't have the genes, you won't be able to produce the protein that you want. So what is gene? Gene is actually a fragment of a chromosome that comes with specific function. So we have genes that make 2 eyes, we have genes that make your hair, we have genes that make your 2 feet, we have genes that make your nose. So basically, if you notice, like Dr. Yusri and me, like we all have 2 eyes, we all have 2 nose, 1 mouth and 1 nose. So basically, our genes is probably 99.9 exactly the same. But we might have some differences, like our fingerprint, for instance, because the pattern of our fingerprints is not exactly the same. So the genes that controlling the patterns of fingerprint might be different. So if I want to know the difference between Dr. Yusri and me, I can actually look at the genes that control the pattern of fingerprint. Then I know that there is a difference between the 2 of us. Okay, I hope it is not complicated. But then I wanted to talk about RNA, because nowadays, when we detect virus, we are talking about RNA all the time. So, but what is RNA? So basically, genes contain all your genetic information. Basically, it's stored there in your nucleus. And then when you need to use it, it's almost like a master copy. And you keep it there to use. So when you want to produce certain protein, what you do is you don't use your master copy, but you copy a section of your master copy to produce what you want. So for instance, now I want, like I eat a lot, so I need to gain more weight. I need to produce, I need to make more fat. So the sections of gene that control the production or the making of fat will be amplified. And then from the master copy, they're gonna copy it into a copy of the master copy, and that copy of the master copy will be used in the future. So basically, the copy of the master copy is the RNA. So for instance, if I just want to make certain protein, I only amplify my RNA, the gene into that section, into RNA, and then use that as a template to produce the protein that I want. So basically, this is the process. Okay. That RNA is called mRNA, but never mind. So basically, this is the whole process. So you have your nucleus, and then you have your DNA. If you want to produce certain protein, you're gonna make RNA as the template, and then send it to ribosome. Ribosome is like a factory, which is gonna be the place for you to produce the protein. So you have the template, which is the RNA, and the ribosome, which is the factory, but you need a building block. So the building block is what we eat. What we eat, everything that we eat is gonna be broken into single amino acids, and it's gonna be carried by tRNA. So basically, the building block is what you eat, and it's gonna be carried by tRNA, transfer RNA, and the cast or the mold, the template will be the RNA that you get from your gene. So you need to have the gene, and you need to have the food that you eat in order to be able to produce the protein. Okay, so that's basically synthesis of protein, very basic. Okay, but then how do we use DNA for environmental forensic? By the way, this is our student Nithya, our master's by coursework in environmental science, and that's Kasrina from Indonesia, and I think that's Atika, but I'm not sure. Okay, so basically, for example, synthesis of protein. So you can only produce certain protein if you have the gene. If you don't have the gene, you will never be able to produce it. So in this case, for alga bloom, some alga bloom actually produce toxin, and the toxin can kill. I think we have recently, a few weeks ago, I think red tide in Penang. So you have fish kill, but sometimes you see the fish is dead because the algae produce certain chemicals or toxin that can kill the fish. But sometimes the chemical actually doesn't last in the environment for a very long time. It will break. But then the gene that the algae has will stay there. So if you go if you see a fish kill and then you go there and you don't see the toxin anymore, you cannot say that, okay, there is no toxin there. But if you look at the algae, you know that this algae is capable of producing the toxin. So you can then relate the contaminations into the algae because you know that the algae has the genes. I don't know if it sounds very complicated. I hope not. Okay, and then also this is hypervariable region. Okay, so every animals or every so basically, like animals normally they have, like chimpanzee and human, we almost have like 90 or 98% similar genes because, like I said just now, we have 2 eyes, we have 1 nose, so basically they are very similar. But there are regions which is hypervariable. These regions are different, like our fingerprint, for instance. So if you want to know, like for instance, in the case of COVID-19, coronavirus, you can actually find genes which is specific to that species only. So you know that this genes only coronavirus has this gene. So if you detect that genes inside your samples, you know that you have coronavirus. If you don't have that genes, that means you don't have coronavirus. So that's how we use genes or DNA technique to actually detect presence of some species in the environment, or in the case of coronavirus in human or in wildlife. It's similar, same technique, but different application. Finished. Okay, I already talked about this. Yeah, so basically that's why you actually have been listening to molecular testing. So what they do is nasal swab, and they're looking for this SARS-CoV-2 is actually gene specific to coronavirus. So they're going to amplify the RNA. The reason they call it RT-PCR because it's actually reverse transcription RNA, the PCR polymerase chain reaction. But I think it's going to be complicated. But what they do is actually they take your sample and they find gene specific to coronavirus. Once they find that gene which is specific to coronavirus, they know that you actually have coronavirus. This technique is actually very, very powerful and can also be very good because actually you can actually, like, sometimes you can mix samples of like 10 people, and then if you don't have any, you don't find any gene because it's very efficient. Within the 10 people, you don't have to test one by one. And if you find it, then you can actually test one by one and see who has the coronavirus. So how do we do it? For instance, we normally collect the samples, nasal swab or anything that contains your gene, and then we're going to extract the DNA or the RNA, and then we're going to extract the DNA. The good thing about DNA is actually precipitated in alcohol. So basically we're going to mix, we're going to take the samples and we're going to break them, and then you have a soup of DNA and protein, or RNA and protein, and you're going to remove the protein, all the other debris, and you get the DNA, and then you're going to amplify the RNA, the DNA, and then you're going to get the gene and compare it with the gene bank, and then you know that what the genes are. And for that reasons, you know what species is there, or what protein, what genes does it have, and what can it produce. Because again, whatever that you are able to be or to produce or to encode, they call it, in, or whatever the protein that you can make in your body has to be depending on the gene that you have. If you don't have the gene, you will never be able to do it. Like if you don't have genes for blue eyes, you will never be able to produce or to make the blue eyes. So examples of the research in our school, actually we have this one. We sample like water in Teluk Bahang, and we wanted to know if there is any toxin producing gene over there. We actually found that it's not a very serious problem. It's actually, we thought it's going to be a lot, but actually it's very little, and it's very good. Actually, Teluk Bahang is very good. And we also go to like other places in Malaysia for this gene that produce toxin. For example, anatoxin A, this is from Miri, Sarawak. And we also go all the way to Antarctica. Like this one, I went to Antarctica in 2016 also to find the genes. They have the, because the idea is, you know, Antarctica is not a polluted area. So if you find the genes over there, so I mean, then it might be spread all over already. And this is examples of our microbiology lab, Tina and master students. And this is our forensic lab. We have actually a big group of people from Nigeria, Libya, and Yemen, India, Iraq, and Indonesia. And this is, I don't know if you remember, in 2014, MH17, and actually one of the guy, this is the deputy superintendent of Penang. He's actually currently our PhD student, supervisor me in USM. So he's doing forensic, but his focus is into human, not environment, but the technique is very similar. And this is an example of how you do it. This is Faisal, our PhD student. And this is how do you basically extract the DNA. Oh, I'm sorry for the sound. I'm gonna make it, yeah. So basically, this is to give you an idea that DNA extractions and the whole process is actually not that difficult. It's actually quite simple. So basically, you're just going to pipette things and things, but look at him, like he has to wear the glove because this is to protect the samples from the DNA from his hands. Because if he doesn't wear gloves, then the DNA from his hand is going to contaminate the samples. So basically, you're going to do a lot of pipetting, sometimes hundreds. And I think for the COVID testing, it's going to be tough for them because there is a lot of piping involved, and sometimes it hurts your finger. I think that's all my talk for now. Again, thank you very much for the invitation today. I am really honored, and if you have any questions, I pass it to Dr. Kam. Thank you very much.